Hey, this is Cole Rasmussen, and I'm going to talk about the sliding filament theory. The sliding filament theory is what actually explains the mechanisms of muscle contraction based on muscle proteins that slide past each other to generate movement. Now, the skeletal muscle system is voluntary compared to the smooth muscles and the cardiac muscles, which are involuntary. So in this case, we're going to talk about how the body creates electric impulses through the nerves that is going to make muscles contract, moving muscles voluntarily. Here you have a cross-section of some skeletal muscle, which is made up of multiple muscle fiber bundles. And within the muscle fiber bundles, you have what are then muscle fiber cells. Inside the cells themselves are going to be myofibrils, which we'll get to in just a moment. But I wanted to show a quick cross-section of that. Looking here, this is where a nerve comes down and attaches, or where it's going to then give the impulse to the muscle fiber cell. Now that electrical impulse that is going to travel down through the nerve is called an action potential. And what's going to happen, or just to explain this first, here we have the myofibrils just underneath the muscle fiber membrane, which the muscle fiber membrane is called the sarcoma. This is the synaptic bulb, which is a part of the axon tail of a neuron. And where that axon tail comes and meets the membrane is called a neuromuscular junction. Since this actually does not physically touch, it functionally touches, so that it actually sends messages across the membrane through a space right here, and this space is called the synaptic cleft. So when this is coming down the nerve, the neurotransmitter chemical is released, which is called acetylcholine, or what is otherwise known as ACH, across the synaptic cleft. That is then going to communicate with the membrane and with the T-tubules that are over here that are then going to release some calcium storage, which we will come to in our next slide. Looking here at a myofilament or myofibril, we have what are then our sarcomere, which gives the myofibrils its striated look. And between every sarcomere is what is a thick filament protein and a thin filament protein. The thick filament protein is made up of myosin protein. The thin filament protein is made up of actin protein. Now, on the actin protein are two other protein molecules that are called troponin and tropomyosin. I mentioned before that as the ACH travels across the synaptic cleft and communicates with the sarcoma, it's going to release calcium stores, and those positive ions are going to get traveled through the myofibril. In just a moment, I'll explain that process, but it's going to release calcium. The troponin is going to attach to the calcium, and the tropomyosin structure is going to move, allowing the myosin heads to attach to the actin proteins and create a pull or a sliding process, which is referred to as, this process is called cross-bridge cycling. And that is what creates the contraction of the sarcomere, and this then allowing a muscle to contract, getting shorter. Just want to go over a little bit more of the anatomy of the cell. So here's the sarcomere. Here are these little holes, which are the T-tubules. Inside the tubules is this more netting material that's going to communicate. The signal is going to come through the T-tubules and go across then what is the sarcoplasmic reticulum. I really hope that is able to express what I've learned through this process, and that I answered with everything that we need. This was kind of a fun experiment, actually, or finally fun to kind of really get to learn and see, so I've enjoyed it. And thank you for your time.